What is the relationship between jumping genes, so-called transposons, and other parts of the genome? How can they be useful for the host species, and what is their role in the emergence of new biological species? Are transposons related to viruses? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Kejnovsky, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. Let me remind you that transposons, also known as jumping genes, are pieces of DNA, thousands of letters long in the genetic alphabet, capable of moving within the genome from one place to another. I have already talked about this several times before. A single genome usually contains dozens or even hundreds of different types of transposons, which differ in their structure and mode of reproduction. Transposons that are similar to each other form so-called families. Individual transposon families differ in the sequence of letters in their DNA as well as in the number of their members. We can look at the genome as a kind of ecosystem in which individual elements occupy specific niches. Relationships between different transposon families can range from cooperation through competition all the way to parasitism. Noteworthy is the presence of non-autonomous transposons, that is, defective elements lacking the genes encoding important enzymes necessary for their transposition. They solve this situation by taking the necessary gene products, such as the enzyme reverse transcriptase or integrase, from their autonomous partners, who, so to speak, supply them into the cellular environment. If we consider transposons to be genetic parasites, then non-autonomous elements are essentially parasites of parasites. Thanks to their smaller size, they reproduce more quickly. However, since they cannot destroy their hosts, a certain balance eventually develops between autonomous and non-autonomous elements in the genome. The relationship between transposons and their hosts, that is, the rest of the genome and especially the genes, evolves over time. They undergo mutual adaptation. This is nicely captured by the rule of the 3 C's: conflict, compromise, cooperation. Let's take a closer look at this rule. At the beginning, a new type of transposon enters the genome of a species in which it was not previously present, for example, through so-called horizontal gene transfer. We will talk about this in more detail in one of the upcoming episodes. It causes something like a genomic shock. The transposon begins to multiply uncontrollably. Originally, it acts as a parasite. After all, that's programmed into its DNA. This is the conflict phase. But after some time, the host activates defensive epigenetic mechanisms that silence the aggressive transposon. Specifically, certain chemical groups attach to the DNA of the aggressive transposon, thereby suppressing its expression, its jumping. In the next phase, both sides reach a certain truce. This is the compromise phase. Transposons can even regulate their own activity so they don't cause as much harm. After all, they wouldn't want to cut the branch they're sitting on. The death of the host would be fatal for them as well. Finally, in later stages of evolution, they may even cooperate with the host when the host puts some of them to use for its own functions. In these cases, we talk about the so-called domestication of transposons. Many transposons are related to viruses, retroviruses, pararetroviruses, and Gemini viruses. Some retrotransposons have a gene for virulence, the so-called ENV gene, similar to retroviruses. It remains a mystery whether these are retroviruses that have lost their infectivity or whether, on the contrary, retroviruses are retrotransposons that have acquired a virulence gene. We have an evolutionary stage here, but we don't know which path evolution took to reach it. The boundary between viruses that are able to leave the cell and transposons that are confined to their own genome is very, very blurred. You might be wondering how it is possible that the genome allows such a large amount of material to just lie dormant. Or can it somehow use transposons to its own advantage? I'll answer you with a few examples. In the well-known fruit fly Drosophila melanogaster, which geneticists have used for years as a suitable model organism and which is called the queen of genetics, scientists were unable to find DNA sequence motifs characteristic of the ends of chromosomes, the so-called telomeres. It eventually turned out that retrotransposons are present in the telomeres of the fruit fly. Retrotransposons jump into the telomeres and protect the ends of chromosomes from shortening, which would otherwise inevitably occur with every cell division and is associated with aging. Mobile genetic elements were even present at the origin of the vertebrate immune system. The formation of antibodies actually involves special DNA rearrangements, whose mechanism is very similar to the movement of transposons within the genome. Even new cellular genes can arise through the recycling of transposons. It's also curious that retrotransposons are responsible for the striking coloration of wine grapes, both red and white, or for the wrinkled appearance of pea seeds, one of the traits studied by Johann Gregor Mendel. Of course, he had no knowledge of transposons at the time. This raises the question of whether the dynamics of transposons can also play a role in the emergence of new species, that is, in speciation. Apparently, yes. By determining the age of individual transposons, it has been shown that in the vertebrate lineage, explosive amplifications of transposons occurred precisely during periods that preceded the separation of new evolutionary branches. What if it is precisely the activity of transposons, triggered by changes in the external environment and the resulting genomic instability, that underlies the evolutionary plasticity of a species during speciation, as Professor Jaroslav Fleger compellingly describes in Frozen Evolution? Today, it is therefore beyond doubt that transposons are not just parasitic elements in our genomes. They are genetic entities that significantly increase the evolutionary potential of their hosts. They help us survive and adapt to changing environmental conditions. Sometimes it even seems as if transposons are reshaping their hosts in their own image, as if they are shaping the entire genome, including the genes themselves according to their needs as a place for their own survival. But in reality, it's a beautiful example of the logic of evolution, which works by patching and mending without a plan. In its creation, it uses whatever is at hand, even if those are parasitic elements. Can we even talk about foreign elements and hosts here? What is ours and what is foreign? After all, our entire genome, including our transposons, is us. That's all for today. In the next episode, we'll talk about genes, what they look like, and how they change over the course of evolution. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. It helps the videos reach more people. I really appreciate your support. Thank you and bye-bye.